So for today's lecture, there's a bunch of stuff to download. You don't need it all right away, but the PowerPoint is the first thing to get. That'll go step by step. So today we are going to identify orthologs using BLAST on bridges 2. And I sort of structured the time so that we'll get our BLAST jobs running on bridges 2, and then we'll talk about why we're doing what we're doing, and then we'll circle back, and hopefully those jobs, or at least some of them, will be done. You can get a peek at what the results will look like, and then I will talk about other approaches to ortholog identification, and we'll end with the instructions for the assignment. So we talked a little bit when I introduced multiple sequence alignment about how when you make an alignment, you make this sort of implicit assumption that the columns, like the sites, share evolutionary ancestry. Those are homologous sites. The next level of that is that you assume that the two genes that you're trying to align are orthologs of each other. And we'll get to what's the difference between orthologs, paralogs, homologs in a second. I know there's a lot of jargon there. So basically, you have to assume that what you're aligning is the same gene in the two different species. And so that's called an ortholog. And so what we're going to go through today is how do we find, or how do we define the orthologs when we've got whole genomes worth of information? So the assignment is going to be to infer the comprehensive set of, and they're always putative in the sense of I don't have a time machine. So it's always a hypothesis about the orthology. And so we are going to take all three of these beetles' genomes. Why beetles? 1, because I work on the bottom one. 2, because it's a happy medium between giving you bacteriatoid data sets and giant vertebrate data sets that are slightly more problematic even than insect or arthropod genomes. And those are way overgeneralizations, right? There are some huge, gnarly arthropod genomes out there too, but these are not so bad. So normally what you would do is, we talked about this in the workflow for multiple sequence alignment. Where do you get your data from? So you could go get the proteomes for each of these 3 species from NCBI. So I've sort of given you the little screenshot click here if you were going to go get it from NCBI. But one, to save time, and two, so that we're all working on exactly the same version of the annotation, because these annotations are constantly being updated. Like we're going to work on tribolium-caseum 5.2, which is the best gene set, but it has been updated since last spring even. So just to make sure that we're all on the same playing field with same starting data, I have put the protein files in Canvas. So that's the next thing you need to have downloaded from Canvas. So the protein sequences for RBBH, you need to get all three of these. And then once you've got those, you need to move them to Bridges 2. So you can do this from maybe, there we go. So I'm dropping out lots of intermediate instructions here. We've done these things before, so if you don't remember, you'll just have to go back. For this data, you can probably run this in your home directory if you want to create a folder there to house everything, because we're going to get a proliferation of files, and it will get messy if you just leave it in your home directory. So I suggest either creating a folder here, and so you can do that by clicking that button right there, new directory, or you can go to your Oceans home directory and do that. As long as you know where it lives, it doesn't really matter where you run it for this example. So I know these are not huge files, but it may take a second, so pause while you get that going. It's important before you leave today, you can execute the SLURM job. So raise your hand if you're having issues at any point. If you've finished moving the protein files up there while you're waiting, the next thing you're going to want to move are these SLURM job scheduler scripts. So even though you don't know what they do yet, you can download those and put them in the same directory with where you just put your proteins. Everything going okay? Yeah. So if you don't get any errors and it doesn't show up right away, you can refresh the page, right? So that there's a refresh button there that, if nothing popped up, but it also didn't tell you you had an error. Sometimes it just, the directory hasn't refreshed. We also may have a bandwidth bottleneck if you're all trying to move 30 megabytes. What? The what? Yes, there are 3 protein files and 3 SLURM job scripts. 3 that end in .faa and 3 that end in .sh. Just curious, does anybody's on-demand browser window still look like this? I just now realized that they changed the interface. Doesn't matter. It'll be small enough that it'll fit in the home directory. You can always move the whole thing later if your home directory fills up, but the only thing that does matter is that those job scripts are in the same folder as the protein files. Yeah.
I downloaded all 6. Only 3 came in. Do you think it'll mess it up if I try to download the other 3? Or, like, I don't
Only 3 came in.
Like, it just stopped downloading and it gave me these, and I refreshed it. It took them all away, and
So
then it gave these back.
that looks good. So the job scripts didn't upload.
No, but I'm just worried if I try
Okay.
to download again, it'll be like a weird duplicate situation. But don't worry.
Hit refresh. So use that one.
Oh, good. See, it takes a long time and then it gets them back.
Yeah. So try doing the job scripts again. They're not in that folder.
Sounds good. Thank you so much.
Yeah, when I was doing it, but it like it only did not do them in the app. But I didn't have to re-download them.
Just doesn't mess it up. I don't think I
Oh, yeah, you're yeah.
I don't know why I did that, but it like stopped after getting through them.
Thank you. Thank
Yep.
you too.
Everybody else got all 6 of them up there?
I didn't do correctly, so
Yeah.
next time I log in, should that appear on the side?
No. Those 3 are the only ones that are ever there. It's really inconvenient that that Oceans one doesn't get to your home in Oceans either. It just goes to the Oceans top level. There is a way to make a shortcut, which is what I have on mine, but I can show you how to do that another time, but it's not needed, so I haven't gone over it. Okay, next step. All right, so once you've got, you actually don't need the job scripts up there yet. So first we're just going to act on the protein files. We're going to make them blastable. So we went over on Tuesday the BLAST heuristic. In order to make BLAST run really fast, one of the things it does is some kind of cool indexing for all those words. So like it can search words really fast. And then the sort of nearest neighbor indexes and things like that. Those are sort of pre-computed in a way so that it can look through genomes or proteomes really fast. But in order to do that, we've got to index it. So start an interact session. So you'll have to start the shell, right? So from dashboard, you can go straight under clusters. There's a button, or you can go back to the on-demand dashboard. Okay. Log you in. Yep. So here, there's a drop down for clusters, and then the only thing there is shell access. It's just a shortcut. You can also, right, if you go back to your main dashboard, that button right there does the same thing. Okay, so what I'm doing right now is struggling to find my directory from the command line. Man. Because I have mine set up on Ocean. Oh, it's M06. Okay, so I had to get into the directory, and now I'm going to start my interact session. I could have started interact before that, but I would have just wasted 5 minutes on. So hopefully everybody can get on interact today. We won't have to be on here for very long. So we only need to be on here long enough to get our BLAST databases indexed. So step 2 in making them blastable is we run the module load BLAST command. Software that's pre-installed on Bridges 2 are called modules. They're all under modules. Is it all caps? Yes. So BLAST has to be in all caps for this module because everything's case sensitive in Unix. So if you don't get any errors, that means it worked. For example, if I did module load lowercase BLAST, it would have told me module not found. And then we're going to run this command make blast db, all one word. And then it's space dash in and then the file name. And we need to give it the name of one of our protein files. Okay. I forgot the rest of the file. Good grief.
We have enough spaces in the code.
Okay. So I accidentally hit enter too quickly the first time. Forgot to tell it which type database. So make blastdb the input file. I'm just doing it on AGLA here, but you'll have to do it on all 3 of the protein files. Yours has the whole word protein in it, doesn't it? Okay, so be aware. So what I'm just realized is that when I renamed them, or at some point I renamed them before I upload them to Canvas. Mine is called AGLA 2.0 underscore prot. Oh, this is going to cause problems. So yours is called protein, EIN, the whole word. But when you execute that, you should see, it should go fast, but you get a report that says it added how many sequences and how long it took. And if you do LL now all three of mine are already complete. But once you run that on each of those protein files, what you end up with is going to generate 7 new files for each of those species. And those are the indices that BLAST needs to be able to search on.
Is it okay if our total number under where you put the command LL is different than the one on the PowerPoint?
In what way?
So I have 82714, and then this one has 571.
Oh, yeah.
Okay,
That's
so
fine.
this
As
one-
long as it generated all the files.
Okay, because I did this one, and it gave me that. Then I did the second protein, and got that.
Yep.
The third got that, and now
Yep.
I have this.
Yep,
So
that's
then
all
it
good.
should be 7 times. There should be 21 here. Should I get
3. There should be 8. There should be 8 total files for each species,
okay.
because you have the original protein file and then 7 index files. But you've got 3 extras. Those 3 SH ones are the job scripts, so I'm not counting those, right?
Okay,
Yeah.
perfect. Awesome. Thank you.
So, so that command line doesn't have a space before it has CD and then slash right away. You have to have a space after the CD. Yeah, so if you hit your up arrow and then go put a space, just use your cursor. That won't work. You've got to use your arrow keys to get over there. And now just hit space, enter. There you go. Yep, that's what I was just coming to do myself. So once you've generated your index files, exit the interact, so you're not racking up charges. And then if you hit LL, you should see, once you've done it for all 3. Now I've got some extra files in here because I have done runs in the past. But for example, on Anoplophora, the longhorn beetle, I've got all these 8 files now. And those sets of 8 files are what you need in order to be able to blast something. So what you've just done, for future reference, is create your own BLAST database. So instead of going to NCBI and searching against the non-redundant set like we were doing on Tuesday, now you've created your own local BLAST database just on these species proteomes. Okay.
I've been showing some people to use the -H for help on how to do inputs.
How to do inputs.
Like, I don't know, every time I use a command that I haven't used before, I do -H.
To get all the usage information?
Yeah.
It's okay. How are we doing? Anybody not indexed yet? Okay. Errors or just Okay.
When we indexed, you want to stay in the BLAST directory for your the next task, right?
Yep.
And if you loaded like the BLAST module once, you have to continue to do it even after you're out of it.
So first thing you'll see in all the SLURM scripts is module load BLAST.
So it won't remember.
No. As soon as you exit the interact, anything, that whole environment goes away, just like on Colab. Maybe not just like Colab, because the files still stay there. Like the files don't go away. So like you notice, you created all those index files in the interact session, but then when you exit interact, they're still there. In Colab, that doesn't work, right?
I meant to show you something.
Okay. So I finally got to the blast, and then I did the make blast DB, and then
Oh, yeah, but that's giving you usage information because it doesn't know what you want to do. Oh, I see. Okay, got it.
yeah, and then it says command out.
Did you do module load BLAST? Did you do okay? Oh, make BLAST DB. Right.
I put that, and then it did this.
Right. That's the usage information, but you didn't tell it what to do with that command, and then the next time you did it, you forgot the DB. So just hit your up arrow. Okay, and then use your left arrow to go over there like that. So, and just add the DB on the end of that. Nope. Sorry, I didn't go all the way over there. So right. Rather than have to retype everything, if you're just missing 2 characters, right, now it will work.
Do I need to add the
Oh yeah. Sorry, didn't notice that wasn't there. That doesn't work. You always have to use the arrow keys. The terminal doesn't recognize your mouse. Yep. There you go.
And then the index part. So we just hit exit,
or? So have you done it for all 3 of them?
I have not. So
okay.
For all 3,
Yep.
And then hit the exit.
Yep.
Okay.
And even like just to save you a little bit of typing in time, right? The only difference between AGLA and LDEC is that. So capital LDEC. Oops, yeah, so the DEC is lowercase. Enter. Okay, so that one's done. Now the file name for Tribolium is a little bit different. So you'll have to, right, just modify that name of the protein file so it matches. I think it's TCAS underscore 5.2 or something like that, but I don't remember off the top of my head. Oh. Okay. So I
you have to show you
Okay, so this is the problem I was alluding to a minute ago. All your job scripts have my old file names in them. So the remedy that I think is going to save the most people the most time is I'm going to fix them all and re-upload them to Canvas. Yes, you can rename them yourself if you, like the fastest way would be to use Nano. If you know Unix and you know the Unix editor. Oh, that's right. Yes. I forgot On Demand has that as well.
But if we can't copy and paste this anyway, if we're just typing it, can we just put in the name that we have it as, and it should work?
Yes. So if you're already at this stage where you're getting ready to run these BLAST jobs, we can come back to the problem I was trying to fix in a second. The interact session lets you go back and forth with the machine, but you're capped at an hour, or I think you can request up to 2 hours if you add commands about how long you want to reserve it for. But if you're really going to run big jobs and you need lots of cores, so we were also limited to 64 cores. So how many possible threads can we run? BLAST is the kind of program where parallelization can speed up the job tremendously. Because you can think, if I have 1,000 proteins and I want to search the database, I shouldn't really have to, in an ideal world, wait and run one, that one finishes, run the next one, run the next one. If I can move that to do 1,000 at the same time, I've sped up my job by 1,000 times. So that's what running from this SLURM scheduler will allow us to do. So if you remember back when I first introduced HPCs, we talked about what is bridges. I want to Sorry. I'm trying to hide this thing so that I can blow this up. Too small to see. Right, we talked about how we have allocations on the regular memory nodes. So on a regular memory node, there are 64 cores, and each of those can do 2 threads. So we have a potential to run 128 threads at a time. That's like, think of that analogous to, we could do 128 blasts at the same time instead of doing one at a time over and over again. So if you were having to write these SLURM scripts yourself, you would have to know which partition on bridges do I want to send this to. So that's where the RM comes from. Then these are common parameters, and I'm just giving you the table there from the bridges user's manual. These are pretty consistent across high performance computing that uses SLURM as their job scheduler. And I guess why make it so complicated to run a job on a high performance computer? If that's not a question occurring to you, maybe after trying to write a job script yourself, it will. But that's because there are so many people trying to run jobs on this machine, right? right? So it needs a way to prioritize. So you're going to submit this job to a partition and wait for those resources to become available. So these are the kinds of parameters that say how many nodes do you want? That's essentially how many computers do you want? How many total tasks are you going to run per node? Stuff like that. Okay. I've already done this naming for you, but the top section, and you can open one of these, you've downloaded them, they should be in your download folder on your computer. If you open it with a text editor, you should be able to see what I'm talking about here. So on each of these lines, it's kind of weird that the SBatch instructions start with that pound sign or the hashtag, because normally that means that it's a comment and don't read this line. But this is a unique thing about SLURM, that it reads that part, and then the next hashtag is like a normal comment. Okay, so this is saying name this job, the -J, and again, if you go back to the table, right? So this is what I'm doing. These options, I'm giving it the job name, TCAS versus AGLA. So I know that the outputs from this job are all going to have the name TCAS versus AGLA. So I know which BLAST was being done or which outputs this belongs to. The -O is to name the output file. This is an output log. The -E is for an error log. Okay, so this is things that normally, if you were in an interact session, would be going to the screen. You're not going to be interacting with the screen, so it's going to send them to these files. If you get a job fail or have a job fail, the first thing you should always do is go to that -E file, the error file, because that's where it's going to write the information that it would have sent to the screen to say what the error was. Okay. So if you email me or message me on Teams and say, hey, my jobs keep failing, the first question is going to be, what does your error file say? Okay. And if that doesn't answer my question, I'm going to say, what does the output log say? Because the output log is going to be whatever the program is. In BLAST, it's going to sort of be giving constant updates on its progress. And so you can see where it died. Okay? So sometimes that'll tell you what did it choke on that made it fail, okay, if the error file isn't super clear. Okay? Next thing, which partition? So we're running on RM. Already talked about that. Within RM, we are going to ask to reserve one node. That's the capital M, N, capital N. And then the lowercase n is how many tasks do we want to be able to run in parallel. That's the number of threads. So in this case, we want to use as many as possible on a single node. So the max is 128. And then it asks, how much time do you want to reserve it for? So we're going to reserve it for an hour. That's more than enough time if we're running 128 threads for one of these BLAST jobs. The nice thing is, unlike Interact, which if you leave it on, it just keeps charging, as soon as the job finishes, it will release the resources and you will stop being charged automatically. Okay, so even though we reserve for an hour, if we only use 20 minutes, we're only going to get charged for 20 minutes. If you've never run a job before, sometimes this can be kind of hard to estimate. And this is why a lot of computational, if you're reading bioinformatics papers, they'll give you runtime estimates and stuff like that. That's so you can estimate things like this. Because if you try, I think the cap is 48 hours. You can reserve a node for 48 hours, or however many nodes you want. But if you need 5 nodes for 48 hours, you're going to wait in line for a long time for those resources to become available. So knowing sort of optimally how much resource do you need will help you be prioritized in the right way. If we're only asking for one node for 1 hour, we shouldn't have to wait very long, okay? And then the last things here, in your scripts, I wrote username at something, but in the past I forgot to do that, and so I was getting emails for everybody's jobs. So you'll have to modify that so that it emails you when your job starts, if your job fails, when your job ends, stuff like that. And then the mail type, you can ask just to get it when it starts, just to get it when it ends. In this case, it's going to email you for both, basically. Anything that happens, it's going to email you. It's just a formatting issue.
On slide,
Yep.
do we need to do
Here?
we don't need to type the purple stuff into it, right?
You shouldn't have to type any of this stuff because you've downloaded these.
Oh. Do we have to okay.
Hmm?
Do we have to open the terminal for the SLURM jobs, or
Nope, the terminal you're in is just fine.
so we should not buy this?
Yep. So that top section is all the resource allocation information, like which computer and how many jobs do I want to run at the same time. The part below that are the actual commands that you want to run. And so all three of the files start the same way, module load BLAST, because if you don't load the BLAST module, it won't know what to do with the BLAST P command. Okay. So then those next two lines, and all three of your files have two lines, and I'm just showing the example here for TCAS versus AGLA. It's first going to blast the tribolium proteome versus the AGLA database. And then it's going to search using the AGLA proteome against the tribolium database. Okay. So again, I'll talk all about the how and why we're doing what we're doing. Just a little bit more detail on what those commands are all about. You could go to the BLAST user guide. So NCBI has a command line user guide for BLAST. And this is the breakdown. It's split across two lines on here, but in your file, it's only on one line. So the lines down at the bottom of this slide match up with this more generic version, which is saying you give it the query file name, the database. Now, naming the database, that's just the name of the protein file. Okay, so like TCAS versus AGLA there. And then there are multiple formats that you can get your output in. I'm giving the tabular. 6 stands for tabular. I don't expect you to remember this. I'm just trying to tell you what it's doing. You would go to the manual, the BLAST manual on NCBI if you're going to have to change any of this. And in this tabular format, it's going to give you 3 columns: the query sequence ID, the subject sequence ID, that's the one from the database that it finds, and then the E value. So how good was that match? And the max target seeks, we're just going to keep 1, because the objective here is reciprocal best BLAST. It can't be a reciprocal best if it's past 1, okay? So we're only going to keep the best hit. And then the number of threads that we're going to run 128, and then our output file name. Okay. Any questions about the content of the BLAST job? And I mentioned this when I was talking about MAFT, how we ran MAFT from the command line, but if you went to the GUI for MAFT online, how it had all those click boxes. Okay. If you go to NCBI BLAST like we were on Tuesday and look at the BLAST terminal, there is a sort of, It's a little bit more obscure, but there is a mapping of all the options you can pick on the BLAST page to what you can specify on the command line here. So just in terms of thinking about what's going on between those two different interfaces. What's going on behind the scenes is all the same. Yeah.
Would you be able to, on your shell, do this step 3 so I can see how it's supposed to look? Because I keep putting in what I think is the right command, but it's not giving me anything.
So are you doing SBatch? Are you already to this step?
I'm trying to work through it, but I think I was confused. I thought we were typing in the full command. No. So like this, I don't need to be doing.
So the next step, if there are no questions about BLAST command and what's in the scripts. So if I go, I'm in the directory where I've got all my BLAST index files and everything, and you should have, I'm going to do less, right, to actually look at something. Job. Oh. Okay, so all I did is print the content of one of those .sh files to the screen, just so you could look at it. And you can see that those lines at the bottom are wrapping just based on the size of the window. It's actually all one line. But all that stuff that I talked about is all set up. I just realized one other thing here. Okay. Your file will say your it will say your username or something like that, your email at. Now, this is where the other thing is that these say prot, and that's not going to work, right? Because that's not what your file names have. So, come on. There we go. The text editor in Unix is called nano. So you could do nano job underscore and then the name. And that will open up an editor like this. Okay, it only works with your arrow keys. So it's pretty clunky, but you could go in here and now you can edit this to whatever your username is. You can also go down and edit so that these match your file names. Okay. You could also just do this editing on your own computer and re-upload them. So, yeah. I typed nano in the file name. Once you're done editing, so all the instructions for nano control are at the bottom. So you'll do Control X to exit. Do you want to save it? Y for yes. And then the name. I'm not going to change the name. I just hit return. I did not do it in less. I exited less and then I did nano. Nope. It's going to try to send it wherever you tell it.
I did up to get the last command, and I pressed back.
And so,
And it's empty now because we dropped finish.
okay, because different ST stands for city.
Oh, you said it's empty, like there's no finish. I understand. Yeah.
ST is stop.
ST is static.
There's no underscore there.
So that's if it says R, it's running. If it says pending, it's pending. If it says CG, it means it's canceled. Complete is just
So that was my first job to
double check, make sure your output is there.
I didn't see that. Your nano job first station.
Great.
And then your is almost the same.
Well, yeah, you should underscore. I don't know. Yeah, you're good.
And then you look for like.
And then I'll show you that.
Yeah, I look.
So in Nano, it's a little bit like you kind of have to like look over your flows. So I'm just gotta go to the end. You can't go down to all of every house. Arrows. I'm just gonna show you how to do this really quick because Nano can be a little like the way that it is. Just a little thing, and then now.
I think we're just changing the name. And it says Thomas, but for any of the
It should be really fun.
yeah.
That's the first one. So now you're gonna do save modify buffer. Do you want to save the changes you made? You're gonna shift Y and then you're gonna present. So now you can go in. An easier way to edit this. Nano is really great when you have changes on it, but you can also but this is more like switch file where you just click and see
Just a second. I'm fixing these for everyone.
Yeah, let me just show you how to run it. So job, where this is my
I'm well, I'm kind of curious. So I did this. I did that.
that was great.
Man,
Yeah, that's all I really do. It's whenever we have to do like this, we don't know certain ones. Because now I'm like, I didn't either. I thought we were. I thought it was just the way it was displayed was like easier to see.
But
So I literally was just I was typing it here so that way I could just copy.
So you throw
Yeah.
okay, what's up?
Okay, so I followed the commands all the way to like uploading each of the proteins,
Mm-hmm.
but then it said to exit
Mm-hmm.
the last one, which I did,
Mm-hmm.
and it
So
was
you're
revoked.
back on a login node. That's good.
Back on the login node.
Yep.
Where do we go from there? Because I tried this command and all it showed me was nothing.
Do LL.
Like that.
Yep.
Just that.
Yep.
Okay.
So you need to go to the folder where your that stuff is.
Mm-hmm.
So you need to go to the folder in the terminal where that stuff is.
Home directory. It's in this
Homology inference. Yep.
Yeah.
So you need to go to that directory in the terminal.
Okay, so that would be
CD.
yeah, the CD. Would it be this little line
Space.
CD?
Nope, just space and then capital H tab. Enter. Okay, now if you run that nano command, So could you use your up arrow till you get to the right command.
Oh, right there.
Yep. Now enter.
Is that entered right? The rm.sh?
I think so. Oh, so you don't have it in there yet,
Okay.
I think.
Well, I uploaded the jobs when I was uploading the proteins, but it never showed anything. So.
Yeah, so those jobs aren't in there.
Try uploading them again.
You need to get those .sh files into that directory.
Okay. So just upload the jobs again?
Yeah. So just so you're aware, the job scripts in Canvas have been fixed now. The file names have. Still going to have to change your email address so that it emails you. But those job scripts, they have the right protein file names now.
So is what you just talked about there, what's going wrong for me trying to run the SBAT, and then messes that.
So do LL. Enter. Yeah, so you don't have the .sh files in there. I don't know if they're not moved up and they're in a different directory, or
So when I uploaded them all at the same time into here, but they didn't appear.
okay. So try it again. Try to upload them again. Yeah. Yeah. Okay. So regardless of where you're at right now, okay, so most people are at the stage where they need to be able to activate their shell script. I'm going to talk about why we're doing what we're doing. I think once you get to that point, right, you do sbatch and the file name, okay, and that will make it run. If you want to check on the status, you can do squeue -u and your username. That will tell you whether it's running or pending. If you ever need to cancel it, you can cancel by the job ID. And so the reason you've got 3 job scripts to run is that you've got to do all the edges of this triangle. Right, so the example that I was showing is doing the back and forth between Tribolium castaneum and Anaplophora glabripennis. Okay. The result of running those should be 6 output files, 1 from each of the BLAST jobs. So each of those 3 has 2 BLAST jobs, so you'll get 6 files of output. Okay, so now more about the why. So hopefully that stuff is running in the background. If not, office hours, or I'll hang around as long as we can. But we need to get all the way to what's the assignment. So you have a question about running it?
Yeah, it's fine.
Okay. We'll get back to it. So 2 kinds of homologous sequences. One, I've already mentioned orthologs multiple times. That's what we want in order to infer things about the evolution of the sequence, particularly as it relates to species evolution. But you're assuming that the orthologs are the same gene in those 2 different species, or multiple species. In this case, we've got 3. So if we call it an ortholog, that means it's a 1-to-1 relationship among all 3 of those species. That's a necessary assumption. It has to be an ortholog in order to infer phylogeny from the gene. I'll show you that in the cartoons of the next couple slides. Paralogs are the other kind of homolog. That's the result of a gene duplication, okay? Hopefully that's not all new terminology. If we look at this in terms of the tree, and I say, okay, how long ago did the common ancestor of 1A and 1B occur? Right? It didn't happen at the speciation event, right? 1A and 1B don't share a common ancestor until way down here at the duplication event. So 1A and 1B are orthologs or paralogs? 50-50 chance. Paralogs, yes. 1A and 1B are the result of duplication, okay? 1A and 2A, so comparing this red dot to that red dot, orthologs or paralogs? I got a vote for ortho. How many people think ortho? 50-50 chance. We got 50-50 hands. It is an ortholog, right? Because the red line is tracing the species line there. So those things have only been diverging since that speciation event. Those are the same gene in two different species, okay? And you could extend that to 1A to 3A, right? So following the red line, that's the same gene in species 1 and species 3. So in this cartoon example, all of the A copies are orthologs of each other. If you compare any A to any B, those are paralogs. So, whoa, what's going on? Right. So to answer the question more explicitly, 1A and 1B, how long have those been diverging? Well, they've been diverging for 50 million years in this example. That's where the duplication happened. So when we get to Orthofinder especially, we're going to talk, they talk about species trees and gene trees. This is the same set of relationships drawn two different ways, okay? And so this is meant to illustrate why you have to have orthologs if you want to infer something about evolution. Because the A copies are evolving across the species, and so are the B copies. But they're, right, 1A and 1B have been diverging for a very long time, potentially. So the divergence between the orthologs is expected to reflect the time since the speciation event. Divergence among paralogs is expected to reflect the time since the duplication event. If we wanted to answer a question about something like, has selection been responsible for a difference between species A and species 1 and species 2, right? And specifically, if we knew that it was involved, these genes were involved, we could compare the sequences of that gene to see whether there's been more non-synonymous mutations than synonymous mutations, as an example. And that would tell you something about whether natural selection's been active. But if you were comparing 1A to 2B, that would not tell you about the divergence between species 1 and species 2, right? Because those two genes have been evolving since way down here at the duplication event. So we've got to be comparing the red lines, not a red line to a blue line. Hopefully that makes some sense. If not, you can go back and think about it some more. Our computational problem is, how do we define orthologs? So how do we decide what's an A gene and what's a B gene in that example? Well, if you just do BLAST, like I take the A copy from species 1 and blast it against species 2 and 3, I'm going to get a mix of the A copies and the B copies because there's some similarity there. They're copies of the same gene. But what you see if you plot the bit scores, and this happens all the time, is that you get this distribution of high quality hits, but then there are a bunch of really low quality hits. And so which of those is an ortholog and which is a paralog becomes the computational problem, right? So these down here, even though if it's a best match, it's not a very good match. The stuff up here probably is. So they're definitely not all good orthologs. Looking at that a little bit differently, so now on the x and the y axis, we've got, so you can think of the x axis as, gene A and the y axis as gene B, or a match of gene B. You'll often see where you get this cloud where it's covering more of the query sequence than it is of the subject sequence. So the query, right, that's the one that I gave it. And that's an example, sort of like what we saw in the P450 genes, where there was a chunk of it that aligned really well, but then there was another big chunk that didn't align very well. And so if I have a short, like if I had a copy of the P450 gene that was only those 400 amino acids that match really good, I would hit tons of other P450 genes. I could not tell which, that would fall down here in this corner where it's covering a big section of my query, but not very much of the matches that it finds in the database, okay? So again, when we're talking about doing this on massive numbers of genes, like whole proteomes that we're doing right now, this becomes a problem of how do you separate those two? This is another way. This is if we look at the bit scores versus the percent identity. You get this group that has high bit score and high percent identity. Those are the good matches. It has a really good score across most of the protein and high percent identity. That's exactly what you're looking for in an ortholog. This stuff over here that has modest identity and low bit score, those are like that part of the P450 gene that had lots of gaps in it. So it was missing dots because it's diverged a lot, or it's just not a very good match. It still might be the best match. It's just not a very good match. And so how do you weed out those not so good matches? Because you can't be confident that it's actually an ortholog. One method that, and this was what people did for a long time, is that you just trim by some arbitrary bit score. So we could just cut it off and say, don't keep anything that has a bit score lower than 4, okay? The problem is, it doesn't get rid of everything. People were using all kinds of different bit scores. And it still left you with this cloud. It just moved the cloud over, where we had these now slightly better identity and slightly better bit score, but still not in the same group with the things that are probably really good matches for orthology. And so people decided, well, the best way then would be to blast A against B and see which one you get, and then take that one from B and blast it back to A and see if you get the same one. That is, by a strict definition, they one-to-one matching. And so this is the algorithm. That's what I was just saying. You would call those reciprocal best blast hits, RBBH, for A versus B. As you extend that to more and more species, you have to do, like we're doing a three-way. You first do the reciprocal best blast hit, and then you have to take those pairwise and make a three-way. As you extend, you get lots of combinations that you have to test for. So it has to sort of go all the way around the ring to be one-to-one matches before you would accept it as an ortholog. It works really well in the sense of it gets rid of all of those low-scoring things. So on the left, those are the original profiles and this is actual data there's a really good blog post that I've linked to before. You can go back and read if you want. But the upshot is doing the reciprocal best blast hit eliminates almost all of those really bad matches that you don't want to hold on to because you just can't be confident that it's, or you wouldn't want to make the assumption that it's an ortholog. Okay, so you're left with just that peak that has the high bit scores. You lose all of that stuff that has low query coverage or low subject coverage of the query. And then you're always going to have some low scoring stuff, but it's gotten rid of all the dark blue down in that corner for identity and coverage. So why not just do that and that would be it? Well, it works really well if you only have a few species. You're going to get to see how well it works with 3 species. But as you extend, you can imagine if you have to do all of these pairwise comparisons, you start to lose genes because you don't get the perfect one-to-one in all of those combinations. It's also especially problematic when you have large gene families. So if you have recent duplication where the genes haven't, the duplicates or the paralogs haven't had very long to diverge, just by chance, BLAST is going to hit A to B and B to A, okay? Because they're just very similar to each other. And that's, in effect, kind of the things that you're trying to eliminate because you can never be confident in those situations that it's truly the ortholog. You have a similar problem when you have really long divergence times. So if you have a really ancient duplication, and so you've got an A copy and a B copy, but one species loses the B copy and the other species loses the A copy somewhere along the line, you're going to get a reciprocal best match, but they're not really originally orthologs. They were evolving as paralogs for a long time. And that's something that reciprocal best blast will give you say these are orthologs, these are reciprocal best matches, but they're really not, okay? And then polyploids, it's a problem. That's where you've got whole genome duplications. So you've got paralogs of everything. And then when you have complex domain architecture, P450s, again, being a good example where you've got the problem of that core catalytic domain that matches a whole bunch of other genes, very hard to be confident in which one is the ortholog. Just to show you an example, this is what your BLAST assignment is really teasing apart. This is what you're going to have to do. These are the individual blasts. This is what your jobs on bridges should be doing right now, or you're close to getting them to do. So you've got these 6 BLAST jobs, right? Species 1 to 2 and 2 to 1. That's one job script. So each column here is a separate job script that you're going to run, one of those SH files. In this cartoon example up here, I've posed the hypothesis, or at least not posed the hypothesis. I've deleted copy B from species 2. So during species 2's evolution, it lost the B copy. Okay? It happens. These are the matches that you would get. 1A hits 2A, but there is no 2B. So 1b, when you blast it against species 2, it's also going to hit 2a because that's the best match for it. Okay? And then the other way around, species 2 only has 2a to blast, so it's going to hit 1a as its best match. Okay? And then I won't walk through each of these individually. Once you get all of those 6 outputs, then I'm just showing you this one gene example. Pairwise, comparing species 1 to species 2, you see 1a hit 2a and 2a hit 1a. So those would be reciprocal best matches. So that's a putative ortholog between 1 and 2. Okay. And then I've color-coded these so you can see species 1 versus 3, 2 versus 3. Then if we want to get the 3-way orthologs, the comprehensive set, you've got to go to all 3. So how do you go from 1 to 2, and then 2 to 3, and then back? We're already at 10:54. I apologize. So the code, this is really what you have to do, is write a script that is going to pull out. This is what your results files will look like. You have to pull out the 2 ways. And ChatGPT, super helpful for this, right? It can do this lightning fast, okay? So don't think, oh crap, I gotta write Python code for reciprocal best matches and we ran out of time. Okay, we're out of time to talk about Orthofinder. It's awesome. There's a Colab notebook that will run it for you, but I've also uploaded the results for Orthofinder. There's only one question on the quiz that will ask you to look through those results and find what's the single copy orthologs that Orthofinder found. I'll go back next Tuesday and talk about what Orthofinder does. But I think it's a straightforward enough question based on, you don't have to run it, and I've given you the outputs. So we'll see how hard that is. All right, I will hang around to try to troubleshoot the scripts running. Don't forget there's office hours tomorrow. Oh, yeah. So office hours for them, not just me.
Sorry for the ambush. I know you said the problem would be checking your output. I don't have a text in my output, so I don't know why I might not have a
What?
text.
You don't have a what?
Like a TXT. Here's where I'm at.
Uh-huh.
This guy.
And so did that run?
I thought so, but I don't know if
Okay.
the proof of it running would
Yep.
be.
So look at the .o and the .e. So go back to your terminal.
Okay.
Do less.
Like LS.
LESS. And then, whoops, so you have to do it on one of those. So it ran, it tried to anyway. And so you need to do less and then LDEC dash.
Oh no.
Query file is not accessible. Okay, so it's because there's an underscore between LDEC and the 2. So if you go back to quit that,
Okay.
and then look at your job script. So let's go nano. That's which one? LDEC Agla.
So just like, the asterisk,
I'll see you later, right?
Yeah.
Also, I know you have a lot of students who want help right now, so I can also just come to office hours one day.
So you see how this is like I'm just changing it so the file names match. So if you looked at your protein files, they have underscores right there. So Control X, yes, enter, and now if you do sbatch,
I tried a lot
there.
of things. I'm sorry.
Nope, that's okay. So that's submitted again now, and it should work if that was the only problem.
Okay, sweet. Thank you so much.
Do you know what I did? Did you
Yes,
follow?
You changed the names
Just-
to match
just-
what I have them as.
Yep.
So I should do that for the other two jobs.
It's just making sure that the commands, the BLAST commands, those file names match what you actually have.
Perfect. If I